Hi, welcome everybody to Andy's BrainCast. My name is Andrew Yan, and I am joined here today with Dr. Amanda Mejia. She is an associate professor in the Department of Statistics at Indiana University, receiving her PhD in biostatistics at Johns Hopkins University. She is the director of the StatMind lab, which develops statistical methods for the analysis of brain imaging data, including principled, statistical, and Bayesian approaches. The goal of these techniques is to increase statistical power and reliability, and also to advance the generalizability of fMRI studies to more diverse populations. Currently, she is collaborating with other researchers to apply these methods to a range of topics, including neurodegenerative diseases, neonatal development, and psilocybin therapy. So welcome, Amanda.
Thank you, Andy. It's so great to be here.
Yes, thanks for your time and agreeing to be here. Now, just to start out with, the StatMind lab, before we started recording, we were talking a little bit about what exactly that acronym means and what the focus is of your lab's research. So why don't we start there?
Yeah, thanks, Andy. So StatMind stands for Statistical Methods for Individualized Neurodiscovery. I actually have the logo here on this fabulous mug that one of my postdocs made for me. And then on the back, I have all of our R stickers that we're so proud of. So StatMind really focuses on the development and the dissemination and the application of statistical models for fMRI and other functional neuroimaging technologies that have enhanced accuracy and power, particularly with an eye towards individualized analysis, so that we can have a more nuanced understanding of fMRI, gain a more nuanced understanding from fMRI studies, but then also be able to start to think about how fMRI can be used in clinical settings. So whether that be a clinical trial or a clinical translation. Of course, a big focus these days is on guided brain stimulation. So using fMRI to identify the precise location in somebody's brain to be able to provide the most benefit from the brain stimulation. And so I think that we're really starting to see some of the utility of fMRI in those clinical settings. And it's essential that we be able to model the data in a way that can be accurate for an individual subject and not just depend on group averages that have been kind of dominant in the field for so many years.
Yeah, that brings to mind a term I hear a lot here in Michigan, which is precision medicine. And I'm sure you must have come across that as well. It seems very popular these days. And yeah, I'd like to talk a little bit about what seems to be a shift in the focus of the field from averages to individualized studies. You mentioned brain stimulation and targeting. There was a paper by the Stanford group about targeting a specific region of the brain to alleviate depressive symptoms. It seems like it's a pretty significant step. Does that relate at all to some of what you're doing?
Yeah, to myself, I have not so far been involved in brain stimulation, but I know people that are, and that is actually something that I'm starting to explore with some collaborators who are working in that domain. So actually last year, I spent my sabbatical visiting Thomas Yeo in Singapore, and he was actually embarking on a clinical trial to look at brain stimulation that's guided by individualized parcellations from resting state data, which is something that he's obviously spent a lot of time developing with great success. And so that trial was focused on treatment-resistant depression. And I think there's a number of trials going on within the United States and worldwide on using brain stimulation in treatment-resistant depression. And there's for a while been a focus on using fMRI to guide the location of those stimulus implants, or those necessarily implants, but the location on the cortex as well. But a lot of the earlier work was based on group average resting state connectivity. And because we, of course, know that people's brains are different and that the location that might be optimal for me might not be the location that's optimal for you, there's an increased focus on using individualized resting state analysis to guide brain stimulation targets in a more precise way, in a more personalized way, as you said, for individual patients.
Yeah, it's a fascinating development in the field. I can't say I know too much about brain stimulation. I haven't worked too much with it, but I do have a lot of colleagues who are running studies, and it seems like
it's an exciting area right now.
It really is.
Yeah, and of course, fMRI historically has been used for pre-surgical planning, and that's specifically task fMRI. And actually, about a year ago, I was looking at some historic trends that I found really fascinating on the clinical utility of fMRI over time. And what I saw is that I was looking at like the number of PubMed publications with the word fMRI and the words that indicated that there was some kind of clinical aspect or some kind of translational aspect. And there was an early spike, like some early promise, some early excitement around the clinical applications of fMRI. And then that really started to fall off in the early 2000s as the clinical trials that were being done really weren't showing any success. And just more recently, we've seen an uptick in in papers that have a clinical focus or a translational focus with fMRI. And I think a big part of it is the analysis of the data. So specifically with resting state data. So with task data, it's the problem is simpler. So we have a known task paradigm. We at least know like we know something about the temporal aspect of the data. With resting state, we don't know what's going on temporally or spatially. So we have to model or make assumptions about both of those domains, both of those things that are interacting to give rise to the observed data. Right. And so the modeling becomes a lot more challenging. And as a result, I think it's more tempting to make simplistic assumptions, like to use group parcellations, for example, to look at connectivity. Just as one example, that really don't lead us to the most precise, individualized insights about a person's brain. So resting states clinical utility has really lagged behind task fMRI's clinical utility. And there's actually even a paper about this that I came across trying to understand the reasons for this. And so I think we're just starting to get to the point in terms of the analytics where we can get insights about what's going on in an individual's brain from resting state in a way that is clinically meaningful.
Yeah, definitely. Resting state is an interesting field. A few months ago, I was talking to Stephanie Noble, friend of the channel, and she wrote a lot of papers early on about how the amount of data we're collecting from resting state is really not as much as we should be collecting. You think about task, you get an hour and a half. In resting state, a lot of people are collecting 5-10 minutes. So obviously, if you're not showing as much respect to it, it's not going to be as reliable. Well, going along that same vein of the applications of some of your statistical techniques to analyzing resting state, can you give a couple of examples of what you're working on right now? And I know from your research, you do a lot of work with Bayesian approaches. And could you talk a little bit about that too?
Absolutely. Yeah. So the areas of statistical techniques that we're working on, there's a few areas that my lab has historically focused on and that we continue to try to develop. And then also, of course, some newer things that we've more recently started to focus on. So historically, I've spent a lot of my attention developing spatial models. That's one of the types of models that I've been particularly interested in. So spatial models try to use the information about the spatial structure of the brain and to avoid the use of massive univariate techniques. So by actually building in components into the model that describe the relationships between locations, we get more accurate estimates. And this can be for task or for resting state. So we get more accuracy and also enhanced power because we're not doing multiple comparisons correction. We're doing a single unified model. So spatial models are something that my lab has had a big focus on, particularly for task fMRI, but then also for resting state. So for like ICA analyses in resting state. And another area, and this has been a bigger focus in the resting state domain, has been the use of population-derived priors in hierarchical Bayesian models but focus on subject level analysis. So the idea there is, say you have a healthy young adult and that you want to analyze. Well, we have a lot of data on healthy young adults, right? We have the Human Connectome Project. We have many other repositories that contain information that we can learn from. And so the idea is that we can establish a prior distribution on whatever it is we might be interested in. So, for example, we've established priors for resting state network maps. We've established priors for functional connectivity matrices between those resting state networks. We can establish priors for task activation patterns. There's a number of things that this idea can be applied to. Our main focus has been on ICA-based and parcellation-based resting state networks and the connectivity between them. That's been our focus. But I think this idea has a lot of applications. So the idea is that you establish a prior based on a larger data set, and then you use that prior in a subject level analysis. And when you do that, you tend to get a noise reduction that leads to much cleaner estimates. But then also, because of the way that we build our models, the priors are designed to respect the data where there are individual differences, so that those individual differences are not just being, washed out by shrinkage towards the group average, but rather the data is being allowed to speak where those differences exist, and the noise is being reduced where there's not really a lot of individual differences anyway. So we are able to get these much more accurate, like, for example, resting state network maps, and then the connectivity between them as well.
I'm really glad you brought that up, because when I, for example, hear about Bayesian techniques, and I'm assuming a lot of people listening to this usually they're much more familiar, and they use frequentist approaches, null hypothesis approaches a lot more. They may have some familiarity with Bayesian, and from my perspective, that's just another way of estimating parameters. But you're talking about a specific principle of Bayesian approach, which is this prior, using previous evidence to more accurately detect what's signal and what's noise. Am I understanding that correctly?
Yeah, so the models that we build could also be built in a more traditional hierarchical Bayesian setting, where you're fitting the model to multiple subjects simultaneously. The kind of key difference in what we're doing is we're actually estimating that prior from a larger set of subjects as a first step, and then applying that prior as a known quantity in a subject level analysis. The key in terms of allowing the data to speak where the differences exist is that the priors that we build are spatially varying. So where there are differences between subjects, the prior is going to reflect that and to do less kind of shrinkage towards the group. Where there are fewer differences between subjects, and it's really kind of like more of a background area, the prior is going to reflect that and to do more shrinkage and therefore more noise reduction. Yeah, but I think I think you're absolutely right that the frequentist approaches are kind of a status quo, right? That's what we all learn about first. That's what the textbooks focus on. Actually, this is the book that I'm currently teaching from in my graduate level stats course. And this next module we're going into is the Bayesian section. And even in this book, which is a bit old, it's a classic, but it's a bit old, the approach toward Bayesian models is, well, this is a bit controversial. It has, it has its proponents, but it just seems clear that the author of this book at the time that it was written was not fully convinced about Bayesian techniques. And Bayesian techniques have become quite mainstream in, I would say, the past 10 to 15 years, at least within the statistics community, as well as many scientific communities. But because of the historic dominance of frequentist approaches, that really remains the dominant approach within neuroimaging analysis and within many other fields. And most of the existing software is focused on frequentist approaches.
Yeah, there is a lot of inertia, so to speak. Now, just to get your personal feeling on this, if you had it your way, would you prefer everybody adopts a Bayesian statistical framework to estimate priors, to estimate parameters? Is there still good arguments to be made for applying a frequentist approach to statistical problems.
Mm-hmm. Absolutely. So I think that it would be incorrect to say that it's a Bayesian versus frequentist question. So there are good and not so good frequentist models for a particular setting. There are good and not so good Bayesian models for a particular setting. So it's not so much about frequentist versus Bayesian, but what I'm a firm believer in is building models that respect the nuances of the data. And I think that the issue that some of the classic statistical approaches for fMRI have had is that they are overly simplistic and they don't really respect the nuances of the data. And so an example of one of the models that we've worked on that tries to respect the nuances of the data is a spatial Bayesian model, where we're not treating every location as an individual entity, but rather we are treating them as a multivariate entity collectively. So that's one example of how a Bayesian model can be used to respect the nuances of the data. But another example is that when we fit a regression model to an individual subject level fMRI scan, there's these residuals, right? And in a classic regression setting, these residuals are assumed to be IID. But because we were dealing with time series data, those residuals are typically not IID in a fMRI regression model. And so we can do something called pre-whitening, where we try to get rid of that temporal dependence by pre-multiplying both sides of the regression equation by a pre-whitening matrix. But the techniques that have been used historically for pre-whitening have also been overly simplistic. So there are better pre-whitening techniques or better modeling techniques that we can use to deal with that autocorrelation. And that doesn't have anything to do with Bayesian modeling. That's just building a model that is more respectful of the true nuances of the data and not making overly simplistic assumptions.
Well, how would you recommend, if somebody's interested in this, wants to follow up on this, how do they start to build an intuition for the nuances of the data? What's a good paper to start with, or book, or GitHub scripts? What would you recommend?
Yeah, that's a great question. I'm on the spot a little bit with thinking of papers to recommend, but the first one that comes to mind in a general sense is Martin Lindquist's 2008 paper, The Statistical Analysis of fMRI Data. Full disclosure, he was my PhD advisor, but I have great respect for Martin's work, and I think that his work is very well respected within the neuroimaging community. So I say that, I think, without any problematic bias. And then also a paper that, also full disclosure, I'm a second author on, but very much in a supporting role, that he wrote is called Zen and the Art of Multiple Comparisons. So that's a great paper if one wants to get familiar with the practice of correcting for multiple comparisons in fMRI and some of the nuances of that.
2015, I believe, was that?
That sounds right.
Okay,
that sounds right.
Somewhere around there. Okay.
And then outside of any of my own work or my advisor's work, I really appreciate the two Eklund papers from 2012 and 2016. So the 2012 paper focused on pre-whitening, actually, and modeling the temporal autocorrelation of the data. And the 2016 paper was the cluster failure paper.
Right.
And so I think both of those papers, are excellent resources for understanding what are some of the nuances in the data, and how are the status quo modeling techniques kind of really failing to recognize those nuances?
Yeah,
and what can be done instead? What alternatives are there?
Right. I remember very clearly when that 2016 paper came out, a lot of people were very anxious. Although you drill down into it, and I know they issued a little bit of how should I put it? Just clarification about the extent of the issue. But yeah, it was. I remember it was pointing out issues with how we had assumed the distribution of spatial correlation was being modeled, and it wasn't being done correctly in a lot of cases.
The spatial the spatial patterns and temporal auto correlations was a big focus of that 2012 paper as well, and sort of a related note.
Okay. Yeah, I'll put links to all those papers down below when we publish this.
Yeah. I mean, it's hard to make up to point to a single resource that can really provide a full understanding of the nuances of fMRI data.
Mm-hmm.
I think that one of my top recommendations for any student of fMRI would be to get as much statistical education as you can, because there's a lot of statistical nuances in the analysis of fMRI data. And I think that it is possible to get by in fMRI without really having a deep understanding of the way that the statistical modeling is working, because the tools are so easy to use. SPM in particular, but also FSL and AFNI make it pretty easy to do the analysis even without that level of expertise. And that's to their credit, that's led to a huge proliferation of analyses of fMRI, which is arguably a very good thing. But it also means that you can do the analysis without really understanding the nuances of how that analysis is being done. And so I think equipping yourself with as many tools as you can with regards to how data is analyzed is a no-brainer, really, for anyone who is at that stage in their careers.
You touch upon one of the trends in the field that I've been observing for a while, which is to be a complete researcher, you do need to be up to date with the statistical techniques, definitely. And one of the, not sure if dilemma is quite the right word, but let's say you're a new graduate student and you've been trained deeply in the theory of some kind of cognitive phenomenon, executive function, for example, and you're trained in that. You know how to design the experiment, you know how to make it a good experiment. And then in addition to that, you're telling them, well, you also need to learn a lot of these statistical techniques. And at some point they might say, well, I just don't have enough time if I do all that. Is the solution somehow changing our training or having somebody like you or Jeanette Mumford nearby to double check everything? I mean, but do you kind of see the issue that some people might run into?
Yeah. I mean, as we see it from the other side as well. So I'm in a stats department. I train statistics students, and we always have these conversations, right? Of how do we get our students to have a meaningful understanding of data in general, but also of a particular domain, right? And I think that as a, kind of just speaking, using this as an analogy, the more scientific expertise a statistician can gain, the better statistician they're going to be, especially if they're doing applied work or collaborative work in that domain. That doesn't mean they're going to become an expert in that scientific domain, right? So as another analogy, I have a new grant focused on the development of biomarkers in Alzheimer's disease using these statistical models that we've developed in fMRI. I am not an Alzheimer's expert, and I never will be an Alzheimer's expert, but I'm learning as much as I can given the realistic constraints upon my time. And I think that we all have the responsibility to embrace the opportunity to learn, even if that's a small amount, whatever is feasible within your constraints, to embrace the opportunity to learn about the tools or the domains that have connections to our primary domain of expertise. So unless we're, sitting in a room by ourselves and developing theory, we're all to some degree interdisciplinary, right? And the more that we can learn about those other disciplines that we are relying on, that we're working with, collaborating with, I think the better science we can all do.
I agree 100%. Let me give you an analogy from how I usually teach imaging methods and techniques. I'm trying to adapt to what's going on in the field. And it seems like Jupyter Notebooks are really popular for demonstrating how to use machine learning or functional connectivity. And they're great because they're portable. Doesn't matter what machine you have usually. They're easy to edit. They're easy to understand as you go step by step. And I found that a very good teaching tool. Is there something similar with some of the techniques that you're talking about as far as like GitHub scripts or Jupyter notebooks to train somebody with like a practical data set?
Yeah, so so in a lot of the tools that my group has developed are in R, and I know that's not really the dominant language of choice within neuroimaging. So people have historically used a lot of MATLAB, and nowadays it's more Python. And R is kind of this third, this distant third, cousin. Yep. But I actually think it's growing. And so the tools that we have, we try to have for every tool. The goal is to have for every tool and something that's really similar to a Jupyter notebook. It's for in R, they're called R Markdown reports, basically. And so that basically provides a demo, or in the language of R packages, a vignette for how to utilize the tool. And I think they are absolutely invaluable because they actually provide the code to do to run the analysis, as well as descriptive, sections where you can describe what's going on, and, interpret the output and provide visualizations. In fact, I'm going to be involved in an educational course at OHBM focused on these spatial Bayesian models in 2025. And, we'll be using some of the demos that we've developed in our Markdown. And that just will provide, because sometimes at OHBM, the educational sessions, we don't always actually have great internet connectivity. Like, it's hard for the students to actually follow along in real time. But that way, if they aren't able to follow along in real time, they'll have something they can take home with them.
That's great. Yeah, I was actually just going to ask that, whether you give any workshops about this in particular. So you will be at OHBM in Brisbane this year?
Yes.
Fantastic.
I have missed almost no OHBM since I started going in 2014.
It's a great conference.
I always try to go. It's my favorite conference, and I'm looking forward to being in Brisbane.
Excellent.
Yeah. And actually that, just talking about workshops and educational venues, I'd be remiss not to mention, and I think this is something that you and I have talked about, the course that I'm an instructor for in Utah every year. And so this is an advanced statistical techniques for neuroimaging and genetics course that was developed by my collaborator, Robert Welsh. And, talking about ways that we can disseminate more advanced techniques, I think that course has really done an amazing job in equipping the group of students that come through every year with the ability to utilize some more advanced statistical modeling techniques for neuroimaging data, including but not limited to fMRI, as well as genetics. And one of the ways that is done is through not just having lectures, so not having kind of lecture after lecture and, instructor after instructor come through, but actually having lecture lab, where the students actually get to do a deep dive into the practice of applying these techniques. And the format of those labs is actually that kind of Jupyter notebook style, where it's like code and that you can run, and, descriptions and outputs. And actually the students, I think, get a really good experience from those labs, as well as from just the extended amount of time they're spending with each of the faculty members. And so there's really a focus on the quality and the depth that we go into. So each instructor will spend about like 2 to 3 days giving lectures and labs with the students, and then also be available for office hours periodically throughout the year, and actually even into multiple years for students who have gone through in previous years. And I think with neuroimaging, it really takes that practical application. You can't really learn about how to do advanced statistical modeling of neuroimaging, in a classroom setting, in theory, or going to a lecture and just kind of like observing. It's so, there's so many computational challenges and nuances that I think we really want to deliver the ability for a next generation to use more advanced techniques. We have to provide the educational forums that can equip them to do so.
Definitely. And also just to aid reproducibility, make sure that, the methods are rigorous, that people know what's happening, why it's happening. That's all really
Absolutely.
important. Yeah,
Absolutely.
we've done a lot of
Yeah.
soul searching as a field the past 10 years about that.
Yes. Yeah. And I think that I don't know what's happening in all current graduate programs, but I feel like that is something that when I was a graduate student, there was very little formal training in.
Mhm.
And in our program, I think there's still very little formal training in reproducibility. And it's something that as my lab has grown, I've actually also had to develop strategies for how do I teach my lab members to use reproducible practices? How do we as a team develop a reproducible infrastructure of code and data and paper, our manuscripts, and that's actually an entirely different challenge than having my own individual work be reproducible. And so I think that I'm glad to see that there's a more focus on this and more opportunities for people to learn best practices and reproducibility.
Related to that, a couple months ago, I was talking to Stefan Bowman, who is at the University of Queensland in Brisbane. And he's the lead developer of a package called NeuroDesk, which is a container. It contains AFNI, FSL, SPM, really any kind of neuroimaging software you could want, but it's all in a containerized space. But he's given a demonstration. You can find it on YouTube somewhere, but he goes through kind of start to finish. It's a contrived small example, but still educational, where he shows how you can register it through something like OSF, the kind of analysis he's doing. And NeuroDesk, because it's a controlled environment, you can also easily upload all the data that you've been analyzing and all the scripts and everything. So I think that that's also just making it easier, because a lot of people, the fewer technical steps as far as maybe like uploading things to GitHub, maybe they're not as familiar with that. That can make it more accessible to more people.
Absolutely.
And they might not be as cagey about these open science practices.
Yeah, and I think Brain Life is a good example of that as well. So this is Franco Pistilli's brainchild and
Yes.
pet project. And I think that's also a big part of the vision of Brain Life, is to kind of democratize open neuroimaging science, basically, and to provide a platform for people to do analyses that are well documented and reproducible, and use best practices.
How often do you work with these large databases? Because a lot of people are excited because they think we have so many subjects, we have so much power now, we have so much leverage. What are your thoughts on that? Have you worked with them a lot? What's your feeling about those?
Yeah, I mean, I would say that a lot of my personal work has been using these large data sets, because I don't collect data as a statistician. That's not part of what my work focuses on. So certainly I work with my collaborators who collect their own data, and I love to be involved in analysis of those data sets. But a lot of the method development that I do is validated using large data sets. Okay. So we've used the Human Connectome project. We've used the in previous work, we've used like the Abide data set, which is a consortium data set, an older data set, focus on autism. And then in a lot of my current work, we're focused on using ADNI. And I really appreciate that these data sets and these resources exist. When I was getting my training, it was much more limited. So when I was a graduate student is actually when the Human Connectome project was initially released. And I was lucky that I was at a place where there was a visionary, mentality that this was going to be an important resource, and we had access to it, probably within the first, few months of its release. And so that's always been a big part of my own method development. With ADNI, I am gaining an appreciation for the challenge that processing these large data sets represents, because we've had to do a lot of the processing in-house for ADNI. It also gives me a renewed appreciation for the HCP and the standardization that it undertook, and the processing that they did to make it just easy to use, and the fact that they did ICA fix on the data and provided a denoise version. It's just such a wonderful resource. But of course, if we limit ourselves to just that small population of healthy adults, there's limited insights that we can get about obviously different populations. And so we don't want to limit ourselves to just relying on the HCP and similar data sets, but it just has really been a wonderful resource. So, but it does require infrastructure. It requires somewhere to store the data, and that's been kind of an ongoing conversation at my institution here at IU is who's responsible for storing this data? Is it an institutional responsibility? Is it a PI responsibility? If multiple people are interested in a data set, how can we more effectively share that within the institution? So I think it's absolutely wonderful that these resources exist, especially for people like myself who don't actually collect their own data. But it obviously comes with challenges as well.
It's interesting times in which, people can make a career even not collecting any data. But at the same time, we have decades of experience behind us, and who knows how many millions of dollars we've spent collecting this. If you don't need to reinvent the wheel, why not just use what's available?
Absolutely. And I think that's where I think it's so important that we can do analyses that give us insights in individuals, because many of the data sets that we have are not large data sets. We're certainly not going to do BWAS using thousands of subjects in every analysis. So we're going to want to get insights from a smaller group of subjects. We're going to want to get insights about individuals, or at least be able to understand why there's where's the heterogeneity coming from, even within a smaller data set. And so that's been a guiding principle of a lot of my work is how can we make the most of the data that we have, because we often don't have either the luxury or even the inclination or the desire to collect thousands of subjects. Rather, we might be interested in a very small population. An example of this in my work is a collaboration with Robert Welsh, my collaborator Vincent Koppelmans at Utah, looking at ALS, which is a very rare neurodegenerative disease. And so we had this amazing longitudinal data set that Robert and his colleagues had collected at Michigan, in fact. And it was like 16 ALS subjects, but they had been followed for like years. And the ALS disease process is very rapid. And so to get that kind of longitudinal neuroimaging data for that particular disease was really, I think, a huge effort and a very impressive thing to have been able to pull off. And so even though we had a small population of ALS, and then we had like 22 healthy controls, I want to say, that were age matched, it was actually a data set that allowed us to get new insights about the ALS disease process. So it was because we were able to pair a data set that had been collected with a lot of care, even though it had been collected years ago. It's not the acquisition was not a modern acquisition. It was using very conventional techniques, but using more advanced processing techniques that had been developed in the more recent years, and then pairing that with spatial Bayesian techniques, we were able to get new insights about the ALS disease process. So I think that there's often this debate of, is it going to be better data? Is it going to be better analytics? And I think there's obviously a role for both. So I'm a huge proponent of using the best acquisition techniques that we can use. For example, the multi-echo work that is emerging and kind of growing in popularity, I think has a lot of promise. But there's a lot of data that's been collected that doesn't have the benefit of the modern acquisitions, but that we can still learn from if we do a careful analysis.
That's an interesting story. And that's incredible having that kind of data set, that kind of resolution on a certain population. Just to get your opinion on these, again, going back to these larger data sets, we talk about these quite a bit at Michigan because a lot of people are interested. but they're seen as kind of a double-edged sword because let's talk about ADNI, for example. It's been acquired over so many years, so many subjects, and technology changes, right?
Yes.
And so you may be analyzing something collected in 2008, 2010, in which the MRI technology, the pulse sequences have advanced a lot since then. This might be kind of tangential, maybe not to your area, but how do you, if at all, I mean, how do you address some of those problems where you have data from way different time periods, different scan sequences, different scanners? If this is too far afield, that's okay.
No,
it's just
absolutely.
something we talk about a lot at Michigan.
Yeah, and it's something that we've started to talk about a lot too, because we're trying to use the ADNI to get to make a scientific contribution through these statistical models in the field of Alzheimer's. And so as we have worked on processing the data and analyzing the data, a lot of those questions continue to come up. So, for example, if we're interested in doing longitudinal analyses or prospective, kind of prediction from one time point to another, if the subject has undergone changes, because of course there's a natural aging process, but also potentially has developed NCI or has developed Alzheimer's disease, has gone through that conversion, or has just progressed in their disease process, how do we disentangle the changes that have happened in the acquisition and the changes that have happened in the individual? So it's a really hard problem. And I think it remains to be seen whether we can even answer these questions with ADNI. We're not giving up, but it does present a lot of challenges. And just also the consortium nature of ADNI. So there's a lot of sites that have contributed data. They're from different scanners. The acquisition protocols are not consistent. There's multiband, there's single band. I mean, it is just very heterogeneous in terms of the site acquisitions. But then, of course, there's also heterogeneity from the population. There's these are different sites in different cities. I mean, different cities have different demographics, different climates, right? Different levels of pollution. Like there's so much actually ingrained in these site differences that even if we knew how to harmonize the data perfectly, we might not really know how to harmonize the data perfectly because there are real differences across sites that we may not be able to account for, right? And so there's just a lot of challenges. And so I completely agree with you. I think it is a double-edged sword, and they are a wonderful resource, but certainly I don't think should replace large-scale data collection efforts at individual sites.
Right.
And, it would be great if that data was public too, but even if it's not, if we can work within our institutions to collect and then analyze data that is collected within the institution, I think that's a really important piece of the puzzle. And actually here at Indiana, we have an amazing Alzheimer's research center in Indianapolis, and led by Andy Sakin and others. And they've collected a lot of data and I know that goes for a number of the Alzheimer's, or of course, all the Alzheimer's research centers throughout the United States. And so part of this project that we're working on is going to involve that data as well. So not just relying on ADNI, but also utilizing some of the single site data that is, I think simplifies some of the problems that the multi-site consortium data sets present. And I think that one of the issues with the consortium data sets is that it becomes so daunting to actually deal with all the problems and all the challenges that they present, that there's a temptation to just make compromises, and not fully address the challenges. And then we don't really know what the effects of that are going to be. And so, yeah, I completely agree with you.
Uh-huh. Well, Amanda, I know that we have some limited time remaining, just a couple minutes. We might leave it at that. I would love to talk to you again at some point just about how your research is progressing, what's happening in the field, maybe come back in a year or two, just catch up with what's going on.
That sounds fantastic, Andy. Thank you so much for having me. I'm so glad that you're doing this, and I look forward to hearing and watching the podcast and all the other people that you'll be interviewing. And I think having these conversations, especially in this extended format, is really going to be a great contribution to the field. And I look forward to seeing you in Australia if I don't see you before then.
Definitely. No, I appreciate all of that. And just to recap, there's the R25 in Utah. Robert Walsh is the PI. You're also involved in that. We'll put a link to that below. And also, you will be giving a workshop at OHBM in Brisbane.
That's right.
2025.
That's right.
Okay.
Yes. And the R25 course, this is an R25, NIH R25 funded training course. It admits about 25 to 30 students per year. And the admissions, it's held, at least for the time being, it's held in April every year out in Utah. I think we'll probably stick with that time frame in the future years, but it's always a point of discussion because it's during the school year.
Right.
But the applications for that they usually come out around November or December. And so just keep an eye on that. We'll all be spreading the word on Blue Sky. So if you follow Robert or myself, or I think Martin Lindquist will be probably posting about it as well. He's one of the instructors, Rick Betzel. So, just keep an eye out for it and mark your calendars if you're interested in attending that course.
Fantastic. All right. Thank you again so much for your time, Amanda.
Thank you,
Andy.
We can make this work. Really great talking with you.
Yeah. All right. Thanks again. Have a great day.
Thanks. You too.